Assalamualaikum and hi. My name is Muhammad Izzazam Rosli. Matric number S58C82. My supervisor is Prof. Madia Dr. Hanis binti Muhammad Yusof. For my final year project, I will do my title project is Green Synthesis of Zinc Nanoparticle, Spigelia anthelmia leaf extract and its get properties. For nanomaterials, nanoparticles are particles of a nanoscale dimension, and nanoparticles are extremely small particles with increased catalytic reactivity, thermal conductivity, non-linear optical performance, and chemical stability to the diatomous surface area to ratio. A nanometer is a billionth of a meter. Zinc oxide nanoparticles are one of the most studied materials because of their versatility in downstream application. Zinc nanoparticles are the second most prevalent metal oxide, and they are inexpensive, safe, and simple to make. Zinc oxide is technologically an important material due to its wide range of optical and electrical properties. There are two methods to synthesize zinc oxide nanoparticles, which are biochemical synthesis and green synthesis. Green synthesis approaches are found to be reliable and economic route to synthesize this metal nanoparticle. In other hand, chemical synthesis method lead to the presence of toxic chemical species absorbed on the surface of nanoparticle. Therefore, green synthesis has attracted attention for the synthesis of various metal and metal oxide nanoparticles. So that's why green synthesis need to be used to complete this project. Significance of study. Green synthesis are required to avoid the production of unwanted or harmful byproducts through the build-up of reliable, sustainable, and eco-friendly synthesis producers. The use of ideal solvent system and natural resources, such as an organic system, is essential to achieve this goal. Green synthesis of nanoparticles also aims at minimizing generate waste and implementing sustainable process. In recent years, green process using meet reaction conditions and non-toxic precursors have been emphasized in the development of nanotechnology for promoting environmental sustainability. This can make the result as good as the chemical synthesis. The plant that to be used in this synthesis is Spigelia anthelmia. Spigelia anthelmia is an important herbal remedy for wound infestation, neurologic disorder, pericarditis, and other diseases of the heart that is continuously being used in the traditional medical and homeopathic practice. The reaction between zinc nitrate and leaf extract or Spigelia anthelmia to prepare zinc nanoparticle. Zinc nanoparticle will be used to synthesize the Spigelia anthelmia, and it will be observed which bandgap properties are comparable or not with the zinc nanoparticle via chemical synthesis. Research objective. The research objective for this study are to synthesize zinc oxide nanoparticle via green synthesis by root Spigelia anthelmia leaf extract. Next, to characterize the obtained zinc nanoparticle using thermogravimetric analyzer (TGA), Fourier transform infrared spectroscopy (FTIR), X-ray diffraction (XRD), scanning electron microscopy (SEM), energy dispersed X-ray (EDX), and the last one is UV visible spectroscopy. And the third objective is to determine the bandgap properties of green synthesis zinc nanoparticle. So now we go to chapter 2. which is literature review. Zinc nanoparticles are zinc nanoparticles with a diameter of fewer than 100 nanometers. They have a vast surface area for their size and are highly catalytic. The physical and chemical properties of zinc oxide nanoparticles differ depending on how they are made. Zinc oxide nanoparticles are increasingly being employed in industry products like rubber, paint, coating, and cosmetics. Due to its outstanding biocompatibility, low toxicity, and low cost, zinc oxide nanoparticles have become one of the most preferred metal oxide nanoparticles in biological applications over the last 2 decades. Spigelia anthelmia. Spigelia is a flowering plant genus in the Loganicae family. Synonym name for Spigelia anthelmia is Spigelia frutuosa Lam and Spigelia domigensis Gan. The local call Spigelia West Indian pink root bush. Plant division for Spigelia anthelmia is angiosperm. For chapter 3 is methodology. Chemical and apparatus. Fresh Spigelia anthelmia was collected from Mukri neighbors at Kuala Nerus, Terengganu and identified by a taxonomist at the Department of University Malaysia Terengganu. The chemical zinc nitrate hexide used for the analysis was prepared by lab assistant. Plant extraction is a method for extracting specific components from plants. It is a solid or liquid separation procedure in which a solid object, the plant, is brought into contact with a liquid, the solvent. The plant that used for extract is Spigelia anthelmia. The solvent is then used to solubilize and contain the plant component of interest. The resulting solution is the desired extract. Plant extracts are used to mediate synthesis which is environmentally friendly. Synthesis of zinc oxide nanoparticle. For the method, uh, the leaves of the plant are washed thoroughly with distilled water to remove dust and other particles. After that, the washed plant part is then dried at room temperature. 50 grams of leaf were weighed and taken for analysis. And after that, they were sliced into small pieces and are crushed using a mortar and pestle by adding a sufficient amount of water. The extract is prepared at two different concentrations. Sample A 25 gm in 50 ml and sample B 25 gm in 100 ml to study the effect of concentration variation in sample preparation. The extract was then pre-treated by boiling it for 50 minutes, which softened the cell membrane, followed by filtering using Whatman number 1 filter paper. In order to remove the debris, it is then centrifuged at 2400 rpm for 5 minutes and the supernatant is taken for further experimentation. Characterization. Zinc nanoparticle was characterized by means of UV spectroscopy for optical properties, scanning electron microscopy, SEM for morphology, chemical structure by means of Fourier transform infrared FTIR spectroscopy, X-ray diffraction XRD for zinc nanoparticle pattern, and energy and EDX for determine its electrical properties. The Soldier and biosynthesis form have absorbent bands of 363 nm and 356 nm respectively. The biosynthesized variation has a slightly greater optical band gap energy, 3.25 eV, than the soldier variant, 3.23 eV. Nonetheless, the biosynthesis version had better antibacterial and phytokinetic response due to a faster rate of stabilization of the nanoparticles by the organic compounds, terpenoids, contained in the Spigelia anthelmia leaf extract.